Hi everyone, today I will share how I review the literature to understand any given subject that comes up in my research. I will walk you through a two-step method to explore the literature where we first go down the chain of references and then we climb up the chain of citations. I will walk you through a real case which I recently encountered in my research, and finally I will explain the most common mistake that derails most literature searches and how to avoid it. I have been actively doing research for more than 15 years now, and I constantly need to learn about stuff outside my immediate field of expertise. This is why honing this skill is so important. Not so long ago in my research, I came across a phenotype where all the sperm produced by a mouse strain lost their heads. Let's revisit how I went about reviewing the literature to understand what was going on with our headless spermatozoa. Well, the phenotype I was interested on is the one of the 10.5c null animals. And this is how the decapitated sperm looks like. The question I was interested on was why 10.5c, an enzyme that regulates RNA, is necessary for keeping the head and tails together. The starting point of my literature review is always Google Scholar. So let's ask Google Scholar about headless spermatozoa. Google provides reviews and regular articles all together. Among the reviews, the most closely related to sperm decapitation is the following. The review is well written and gives a broad introduction to sperm morphogenesis, but let's cut to the chase and try to find the reference by searching the word headless or decapitated. Okay, here we have only one reference to decapitated sperm, which is related to the deletion of the gene ODF1. The next step in the literature search is to follow the reference chain to the main manuscript that describes the fundamental findings. In this case, the phenomenon of sperm decapitation in the absence of ODF1. Now we go and read the manuscript cited in the review. I like to start with the intro and discussion because they usually point you to the most fundamental work. If we start reading the discussion, we find that this paper was a follow-up manuscript of the first paper where they show the headless spermatozoa sperm in the absence of the protein ODF1. In this case, we need to take another step down the reference ladder to find the initial work on ODF1. When we read the manuscript cited in the previous article, we confirm that this was the original manuscript describing the decapitation in the absence of ODF1. So this was quite good. We started from a general review, then we went to a first article on the topic of ODF1 decapitation, and then we went one layer down by looking at the reference to the original paper. Once we find a seminal manuscript in the field, we stop going down the reference chain and we start climbing up the citation stairs. Here is how I do it. So the references of a manuscript allow us to go back in time to look for the original references. With Google Scholar, we can go on the opposite direction and check which manuscripts cite our work. So let's do that with the original manuscript on ODF1 decapitation. While going up the citation chain, it's best to start with a good review as well. Here we are looking for a review approximately 1 to 5 years old. We don't want something older because we might be missing a lot of already established findings. In this case, the review Molecular Genetics of Infertility, Loss of Function Mutations in Humans looks quite promising. It is more specific than the one we originally selected when we were going down the chain of references, and this is the primary objective of the literature search so far, is to identify most relevant reviews on the topic. If we search the review for the keyword acephalic spermatozoa, we get this great table where we have a summary of all known cases of acephalous spermatozoa. Well, at this point we have to start threading the needle more finely. After reading all the papers, you have an idea of what the neck of the sperm looks like and how it's formed. Let's have a mini review here for the sake of the discussion. So reviewing the literature, we have essentially 3 types of sperm. We have like the wild type, and this is the head of the sperm that is attached here to the tail. And we have here in the neck like the mitochondrial, very organized, surrounding the mid piece of the tail. So this is the mitochondria, and this is the rest of the tail. And these are our wild type animals. Now there is a first type of mutants where we have like the head as the wild type that is separated from the tail. But if we look at the tail is still very organized, or at least the mitochondria in the midpiece are wrapping around the tail. But there is a second type of mutants where we have the head missing again, but the tail, instead of being well formed, is like bigger. Because of this, people first refer to this type of sperm as small head, but actually this is just the cytoplasm where you have the mitochondria all disorganized all over the place. Looking at the 10.5c phenotype more closely, we see that not only is the head missing, but also the neck appears to be enlarged. While examining the phenotypes of the different decapitated mutants, we look at which ones resemble each other the most. ODF deficient animals have a similar phenotypes with an accumulation of cytoplasm. In this case, we can go to the ODF1 paper and see what new manuscripts since the review was written are out there and have a look at these manuscripts. The ODF1 paper has been cited a lot during the last 5 years, but there is a lot of reviews and articles related to adjacent fields. What I do here is to look at the most recent articles and not reviews on the topic of sperm formation. In all, there might be 7 to 15 extremely relevant papers that you have to look very carefully. You can complete this entire process within a week. The question now is whether we learn anything during the entire process. But how can we test this? The primary goal of the literature review is not to read every possible paper remotely related to your manuscript. The primary goal of the review is to formulate a hypothesis that can be experimentally tested to explain our previous observations. Only after doing the experiment and analyzing the data, we should go through the literature review again. So it's very important you stop after you have generated your testable hypothesis to avoid going down a rabbit hole. For the 10.5c decapitated sperm, we decided to look at the electron microscopy images of our 10.5c animals to understand the nature of the enlarged neck and how the phenotypes relate to that of ODF1. After looking at the electron microscopy images of 10.5c mutants, we observed that they were highly similar to those of ODF1 null animals, and that prompted us to continue in that direction of research. After analyzing the new data, you can repeat the cycle. Of course, now you are more familiar with the field and you should be able to move faster. Well, that's it for today. Please subscribe to the channel if you like this content. It's the best way to make it accessible to people like you in the future, including yourself. If you are already subscribed, please leave a thumbs up. And if you're working on a manuscript, check the playlist on how to write a scientific paper. I will see you there.